I will start off talking a little bit about my stuff. Took some inspiration from the previous session that Steve did a really nice job handling that, and the way he sort of weaved in the topics of the other people. So I feel a little bit of pressure to try to perform to a similar level. So what I will tell you a little bit about today are our efforts in trying to understand the emergence of protocells. And I think that this is a little bit distinct from what many people do in the origins field. We're certainly not the only ones. I certainly would not want to give that impression at all. But you have a lot of people who look at the prebiotic synthesis of the building blocks, or you have people that try to decipher how life began by reconstructing the last universal common ancestor. Or perhaps they're also interested in protometabolism, but that also oftentimes seems to be through the lens of the synthesis of the building blocks of life. And so what we're trying to understand is a little bit this space in between, not exactly at the synthesis of the building blocks and certainly not close to LUCA, but how the pieces could start coming together. And I hope from these sorts of studies we can gain some useful insight into this transition from chemistry to biology. And so we do this basically through two approaches. We try to reconstruct models of protocells, which basically means that we look at what we think are our best guesses of prebiotically plausible vesicles to house our cell-like systems. And we also try to investigate some types of chemistries that perhaps could have supported these early cells. And ideally what we want is we want to put all of this together because we really hope to be able to reach a point where we can have fuel-driven protocells, something that's a little bit closer to what we think of when we think of biology today, where there's a consumption of a food source and you get growth and division and essentially proliferation. I'm certainly not claiming that we're right around the corner for that, but this is the type of things that we're hoping to work towards. And so when you think about the protocellular compartments, again, I don't want to be misleading. There's a long history here of people who are looking at protocells from the sort of pioneering days of David Deamer and Pierre Luigi Luisi. And I think at least earlier today, I saw David's name in the audience. More recently, the Shostak lab has been making contributions here. I saw Suda's name pop up a second ago. So she's out there and has done some very nice work as well in origins. With protocells, we have a session, I think even tomorrow, that's really focused on it. And I even saw Roy Black's name earlier. So there's certainly many people who are giving very nice contributions to understanding of protocells. And when you see the majority of these studies, including things that I have done in the past, the focus is always on fatty acids and hopefully also short chain fatty acids, which are thought to be more prebiotically plausible. And we really love fatty acids. We all love fatty acids because they're believed to be prebiotically plausible. You can find these things in carbonaceous meteorites, which doesn't get talked about a lot. Unfortunately, there's not a lot of effort into how these things could have been synthesized. I wish the organic chemist would spend a little bit more time on that. It would be very fruitful, I think. But we love these things because they form vesicles, and they form vesicles that can do things that you couldn't imagine being capable of achieving without really highly complex protein machinery. This is not my work at all. This is work from the Shostak lab done by a good friend of mine, Ting Zhu, who's now at Tsinghua University. But he showed basically that with just mixtures of lipids, you can get growth and division. And these are the kinds of things that probably existed on this planet before there was life, the things that could house these chemical systems that certainly, I think really strongly think personally, probably many of us agree, would have aided Darwinian evolution and helped, again, sort of usher in this transition to more lifelike chemical systems. The problem, of course, with these fatty acids, to those that work in the field, is that they're simply not stable. And if you're trying to think of things that are prebiotically plausible that could have survived on our planet, it is a bit hard to imagine structures like these having been the constituents of protocells. It doesn't mean that there weren't the right conditions. Again, there's some nice work out there of, for lack of a better word, I'll say additives that were put in the system that helped stabilize the vesicles. And these are things that we should certainly keep exploring. It's also possible that there were niche environments that are a little bit different than the average compositions that many of us rely on when we try to understand what's more plausible. But nevertheless, they certainly are fragile. And this has been the thing that's really puzzled many of us when we try to think of protocellular structures. And so there was this really beautiful paper from the Krishnamurthy laboratory that showed that when you have a mixture of fatty acids and glycerol and diamidophosphate, that there are these phosphorylation reactions that precede and that then give rise to something that we call cyclophospholipids. And so this to us was a really intriguing, plausible alternative to fatty acids. I wouldn't say that these would replace fatty acids in their entirety. As you can see, that fatty acids are one component of the synthetic mixture here. But we would, if this sort of chemistry transpired, we were really curious to know what kind of protocellular structures would emerge, and would these resulting structures be stable enough to survive more reasonable environmental conditions. And so when the PhD student at the time looked at this Duhan Topralak, the very first thing he noticed was it didn't really matter what pH he was testing. When he looked under the microscope, he saw vesicles. And this is not what you typically find with fatty acids. Typically under really acidic conditions, you get oil droplets, and at really high pH, you get micelles, and there's sort of this sweet spot in between, typically between around pH 8, that you get a stabilization of the bilayers that you need to form vesicles. But that's not what Duhan was seeing when he was looking under the microscope. Now, of course, I don't think microscopy is sufficient. You want to know, are these things really robust enough to hold material across a variety of different conditions? And so when Duhan did a sort of straightforward experiment where you put a large sugar molecule that's fluorescently labeled inside of the vesicles, you incubate the vesicles at different pHs, and then you run a column to see how much is retained and how much leaked out, you find that with fatty acids, and here you can see two different compositions of fatty acids, primarily consisting of decanoic acid, because again, that's something that's considered to be more prebiotically plausible, we find that it doesn't really hold much material outside of that narrow range in the center of the pH scale. And so you can also see that with the cyclophospholipids, and these have been mixed with a fatty alcohol that helps stabilize the formation of the vesicles a bit, that it didn't really matter what pH Duhan tested, we had really high stability. So this was promising to us. We have vesicles that are somewhat insensitive to the pH of its surroundings. And so we next tested the cationic or salt, is a better way to say it, the salt composition of the solutions. And so here, if you test really high concentrations of sodium, all the way up to 1.5 molar, which is obviously far beyond what you'd find in seawater, you can see that it does degrade a bit our vesicles. We lose about half of them at 1.5 molar sodium, but nevertheless, they are much, much more stable than what you would see with fatty acid vesicles. So this was, again, quite promising. If he did those same experiments with magnesium, one of those cations that typically causes problems for us that are trying to work with protocells, because fatty acids are really susceptible to the degradative effects of magnesium, likely just interacting with the carboxylate head groups. And if you think about how biology works, there are lots and lots of magnesium dependent reactions. And even here with the cyclophospholipids, you can see that it is highly stable, not perfectly stable, of course, but much, much more stable than what you would see with fatty acids. And what we found really fun was if you start mixing these salts together, which of course is more plausible. You're not going to have a pool of just magnesium, for example. If you have a 0.4 molar sodium, something more similar to what you'd find in the ocean, plus magnesium, you are able to survive the magnesium much better. So it seems like the sodium is essentially screening the interactions between the carboxylates of the, or not just the carboxylates here, they're cyclophospholipids, between the head group of the cyclophospholipids and and the magnesium ions. So we were really excited about this because, again, now we have a composition that seems to be much more stable to environmental conditions, something that maybe we can start thinking about building things with more complex structure. And, if we compare and contrast this to previous stuff that had been done, again, we all love the fatty acids because they have a lot of associated dynamics that allow it to grow and divide in the absence of any sort of highly evolved machinery. And the downside was its instability. But if you look at more biological-like lipids, like the diacylphospholipids that are shown at the bottom, these are very, very robust. They can survive just about all the conditions that you throw at it, but that stability also comes at a cost, which is to get these structures to grow and divide, you need to have more complex, highly evolved machinery, which basically means protein enzymes. And so we're really interested to see, can cyclophospholipids fill this gap? Could this have played a role in the transition between different types of structures that allowed for growth and division in the absence of proteins, but are also stable enough to survive more plausible conditions? And I hate to build a story up like this and then not show you growth and division with cyclophospholipids, even though that's exactly what I'm going to do. We are working on this, and I can say that it is working, but it's not actually ready for presentation. What I will say is that the mechanisms needed for growth and division with cyclophospholipids are a little bit different than what people have done with fatty acids. And that's because we can't feed our systems with micelles. We basically always make vesicles regardless of the conditions that we're working with. And so we wanted to understand, are there ways to induce growth and division by mixing different populations of vesicles? And the short answer is yes. When Duhan was testing vesicles consisting of fatty acids with one chain length and mixed them with vesicles consisting of fatty acids with another chain length, the sort of disparity in the dynamics of those two compositions give rise to shape changes that you can sometimes observe depending upon kinetics, as well as division. And so this is an example of growth and division through these sorts of processes where we're mixing together vesicles of different fatty acid lipid compositions. One of the compositions of the vesicles are really small, so you don't see it by microscopy. So you're only seeing the big vesicles and the sort of, I don't know if birthing is really the right word, but let's say birthing of new daughter vesicles. So that's what we've been working with when it comes to the compartments. I hope you'll see more from us shortly in the literature on that. And we'd really like to be able to start combining these systems with some sort of supporting metabolism. And we have published a little bit on iron sulfur clusters and their potential role in this process, which I'll get to in a minute. But the thing I would really like to emphasize is that when we talk about metabolism, I think we talk about this in a variety of different ways. We're not always really thinking, I think, in our heads of exactly the same thing. And when I'm talking about metabolism, I'm really talking about the really simple, straightforward process of a living organism taking a food source, breaking it down. It's obviously going to release waste in the process. And it's using the energy that's released from the consumption of the food to support the out of equilibrium state of that highly organized structure that is a living cell. And the way biology, of course, does this is it tends to oxidize the fuel sources. Of course, it doesn't have to be glucose breaking it down to CO2. In fact, that's a really quite modern example. It's certainly not how I envision it to have occurred on the prebiotic Earth. But basically, the idea is you're oxidizing the fuel source. You're passing those electrons down thermodynamically favorable pathways, and you're depositing that into common currencies. You're storing the released energy in common currencies, which typically in biology means you're storing that energy in either proton gradients, sodium gradients, or the synthesis of ATP. And the beautiful thing about this that biology does by funneling all the energy down to common currencies is it doesn't really matter what endergonic chemistry the cell needs to survive. It can always sort of pay with the same currency to get that work done. And I think we're going to hear some fascinating work in this session from Matthew Pasek, who has done a lot of really nice work on geochemical routes to phosphorylation. And so I've always enjoyed reading his papers on this topic. And he's been sort of, I think, one of the very first people to look at this important chemistry. So we look forward to your talk, Matthew. So again, this is the way metabolism is typically thought of, or at least when you study it in biochemistry class, we're taught to memorize these individual steps. This is the citric acid cycle that you see in front of you. And there have been several papers that have come out in the last couple of years that have looked at prebiotic analogs of this. And, I think all of that is important. I'm in no way criticizing it. It's really important to know what kind of chemistry was accessible. And of course, that would have provided a foundation for the subsequent chemistry that emerged to support cells. But again, the focus, I think, has tended to be how can you harness these cycles to synthesize things like amino acids. It's always seems for me to come back to this building block synthesis approach, which again is important. Living things have to build their components, but we're more interested in is capturing the electrons that are released during this process. But if you're interested in the synthetic aspects of it, there's another great talk that you're going to hear today from Yamei Li from ELSI, who's going to tell us about the geoelectrochemistry of these organic synthesis reactions on the prebiotic earth. And I'm interested in hearing more about ammonia, and you even have a bit of iron sulfur clusters there. So we're looking forward to your talk, Yame. So again, what we are interested in is capturing those electrons, and those electrons can be captured, at least in biology, by the things that are represented with the blue arrows. And so this is really what we were after. Can we find a metabolic-like system that we can capture those electrons and then deposit that into a common currency? And ultimately, we would like to be able to exploit that common currency to push endergonic chemistry and to give a selective advantage to one subset of protocells over another. And so we did eventually hit upon a reaction. We found that pyruvate can non-enzymatically reduce NAD, meaning we can take a prebiotically plausible fuel source and deposit those electrons, capture those electrons into a component of the RNA world, right? It is a ribodinucleotide. We talk a lot about nucleic acids and the RNA world, and it's always been a little bit puzzling to me that people haven't actually thought about the roles that some of these molecules could have played in more metabolic-like chemistry. Now, we don't only see this type of reaction occurring by NMR, which I think by itself is quite convincing, but we can see that we are actually reducing NAD to NADH by LCMS, by absorption spectroscopy, and by fluorescence spectroscopy. So it doesn't really matter how we look at the reaction, we are getting the non-enzymatic reduction of our ribodinucleotide. We do not know the precise mechanism. All we can say is it seems to be some sort of oxidative decarboxylation, but the exact process is something that is currently under investigation. Now, pyruvate is an alpha keto acid, and every alpha keto acid that we tested was also capable of non-enzymatically reducing NAD. So we are not reliant on just one molecule, but there's a breadth of molecules. Let's say, as long as you have an alpha keto acid, it looks like from the ones that we've tested that this will work. It doesn't work with beta or gamma keto acids, so it does seem to be specific to alpha keto acids. So once you deposit those electrons from pyruvate to NAD to make NADH, you can pass those electrons on to other electron acceptors. It had already been known before that you can non-enzymatically reduce FAD with NADH, and we were able to show that as well. And then we deposited the electrons in a terminal electron acceptor that was just methylene blue. I'm obviously not trying to convince you that that is prebiotically plausible. It was just our place to sort of dump the electrons and keep the flux going. So you can start to assemble non-enzymatic electron transfer reactions. It is obviously not identical to what you see in biology, but you can find that these reactions will occur non-enzymatically. Now, when you look at how these things are done in biology, there are a lot of metalloproteins and metallocofactors involved, and many times they are exploiting, in fact, iron sulfur clusters. And these are things, of course, that we have also worked on in the past. And one of the things that we had found is that electrons from NADH can be put onto iron sulfur peptides, reducing them down to the ferrous state, and you can reoxidize them with hydrogen peroxide, which results in the generation of hydroxide. So basically, what this little electron transfer pathway suggests is if you were to put that reaction scheme or that a series of reactions inside of a vesicle that is impermeable to the protons, you would generate a proton gradient. This is not through the same sort of mechanism that you see with highly complex proton pumping in biology today, but we are coupling thermodynamically favorable flux of electrons to an unfavorable generation of a proton gradient. So we found this to be insightful, but nevertheless, I have to admit, we use POPC membranes. Those are the kinds of lipids that you find in biology today. They're not the kind of lipids that people typically think of when they're trying to think of something that's prebiotically plausible. And so those are also things we're working on, are different compositions of lipids to see if we can identify compositions that would be able to hold a pH gradient. And quite frankly, if we don't, then we know that we've hit a dead end and we have to find another way of harnessing these electron transfer reactions. So thus far, we're pretty excited about where we've gotten to. We found food sources, which are essentially alpha keto acids. We have a way of generating a flux of electrons that leads to a proton gradient. And really what we'd love to figure out now is how can we harness this? How can we use the proton gradient to do something useful, to keep the system out of equilibrium, and to hopefully give a selective advantage to one subset of protocells versus another, which we would really love if that answer led to something like growth and division, as I was showing you in the first part of this talk. So we are thinking about prebiotic plausibility. You know, we're not just trying to play tricks in the laboratory to see if we can push reactions to go and sell it as something that is plausible and exciting. And so, you know, there have been things that we've done in the past where we showed that photochemistry, the flux of UV light, can lead to the desulfurization of cysteine-containing peptides. And that can be a positive thing because it's a way of delivering the inorganic sulfides necessary for the synthesis of polynuclear iron sulfur clusters. But of course, it's also a negative thing because, you know, if that happens to all of your cysteines, then you have no cysteines to coordinate your iron sulfur clusters. So we were interested to see if there were ways to protect cysteines from UV light. And then we found that if you had thiophilic metal ions, you significantly increase the half-life of the cysteine. So you can see this is not increasing it to weeks or months, but it is a significant stabilization of the cysteines. If we add an even more thiophilic metal ion, we can stabilize it even more. And if we add magnesium, which does not bind to sulfurs, you can see that it's almost exactly the same as the non-metal conditions. So that's one way we can think of stabilization, but it's important to me to really point out photochemistry is not a net negative. There's a lot of fascinating work on the benefits of photochemistry, and we're going to hear about that from Karina Kufner right after I'm done speaking. So we really look forward to your talk, Karina. And we're also going to hear more about photochemistry and sulfur. So this is like right up my alley here, prebiotic organic synthesis by photoactive elemental sulfur in terrestrial hot springs, surface conditions, obviously, that would be exposed to UV light. And so thank you for joining us today on Huai Lu. We look forward to your talk. So we've not only looked at degradation of peptides by UV light, we've also looked at degradation due to heat. And one of the things that we noticed is many times when people are trying to get polymerization or at least dehydration reactions to occur between amino acids to give you longer peptides, they tend to place these things at really high temperatures such as 150 degrees. And we've certainly seen this temperature pop up in some other prebiotic syntheses as well. And so we tested what is the stability of a simple peptide with a cysteine GCG at 150 degrees, which admittedly is very high. And you can see it rapidly degrades. We lose the GCG pretty quickly. And if you add thiophilic metal ions, it does stabilize it a bit, but it's not fantastic. And to our surprise, if we do the reaction, if we run the reaction in seawater, we get really good stabilization of our cysteines. And so if you try to figure out what's going on by looking at the individual components of seawater, you can see that it is actually magnesium that is affording this stability. So something about the carboxylates are involved in the degradation pathway. And the last thing I really want to point out is it does matter what type of ligands you're looking at for the iron sulfur clusters and what the solution conditions are. We have a lot of data on that we're trying to compile now and put together into a cohesive story. But these things are dynamic. You can form lots of different types of clusters, and it's really dependent upon your solution or environmental conditions, as well as the peptidoligands that are there to coordinate and stabilize the formation of the cluster. So with that, I'd like to thank the people that did the work. I always like to emphasize this is not the current composition of my lab. My lab is not that huge, but the data I showed was from Duhan Toperlak, Shivaji Bazik, as well as Daniele Rossetto and Yin Hu. And thank you, and I'm happy to take a couple of questions while Karina sets up.